In this video, we will try to learn the cell cycle regulation. Two important proteins, cyclin and cyclin-dependent kinase, are important for cell cycle progression through different phases of the cell cycle. Cyclin D, cyclin E, cyclin A, and cyclin B are the different cyclin proteins which work together in cell progression during various phases. Now we will discuss about the various checkpoints that controls the cell's progression from one phase to the other. The first checkpoint is between the gap phase 1 G1 and the synthesis phase, that is the S phase. Initially, the tumor suppressor retinoblastoma proteins inhibits the E2F protein expression until the conditions are fulfilled by the cells in G1 phase for its progression into the S phase. What are these conditions? These conditions are: here the cell's cytoplasmic content increases, cell's protoplasm increases, cell organelles are produced, and the enzymes which are necessary for the DNA replications are produced. When all these conditions are met, then the CDK4 and CDK6, which formed complex with cyclin D, both these complex results in phosphorylation of retinoblastoma protein. As a result of this phosphorylation, retinoblastoma proteins withdraws its inhibition from E2F, as a result of which E2F expresses itself and it forms a complex between the cyclin E and cyclin dependent kinase 2, as a result of which the cell progresses from G1 to S phase. Now, if there is any DNA damage or if there is any break in the DNA molecule, it causes the activation of p53 molecule. This p53 molecule in turn increases the level of p21. P21 protein gets bind with cyclin E CDK2 complex until the DNA gets repaired. After the DNA repair, then this p21 protein dissociates from the complex and the level of p21 decreases, which causes the progression of the cell into the G2 phase. Now, cyclin B and ataxia telangiectasia mutated protein, which is also known as ATM. What happens normally? Normally, the cell remain arrested in G2 phase until the conditions are fulfilled for its progression into the mitotic phase or mitosis part of the cell cycle. Now, what are these conditions? These conditions are: the cell should have synthesized the proteins which are essential for the spindle fiber formation and the proteins which forms the microtubules. So when all these proteins are synthesized, then the cell gets ready for its progression into the mitosis. So, before its progression, it remains arrested in the gap 2 phase. How it is arrested? P53 and ATM, both these protein activates CHK1 and CHK2 proteins, and these two proteins inhibits or arrest the cell in the G2 phase. When all the conditions are fulfilled, then only cyclin B activates the CDK1 protein and forms complex with CDK1, and this complex, via phosphorylation of various transcription factors, causes the cell's progression into the M phase. Now, another checkpoint is there in the metaphase of the mitosis. CDK and M phase cyclin, they forms a complex CDK M phase cyclin, and this complex is phosphorylated by two proteins. One is WEE1, another is CAK. So these two proteins causes the phosphorylation of cyclin dependent kinase unit. After phosphorylation, this complex is in inactive state. When the CDC25 protein, which also acts as a phosphatase enzyme and removes the phosphate which is added by WEE1 protein, then only this M phase cyclin and CDK complex become activated. As a result, it forms the maturation promoting factor, which causes the self progression from metaphase to anaphase. Further, release of inhibition of anaphase promoting complex APC got an important role in the progression of cell from metaphase to anaphase. On fulfilling the conditions, the APC gets uninhibited. As a result of uninhibition, the APC degrades cyclin B and degrades securin protein via ubiquitination and proteolysis, which causes separase to be free. The separase now becomes uninhibited, as a result of which they cut the cohesin protein and helps in separation of chromatids, which are very essential because these chromatids now migrate to the two different poles in anaphase. So in this way, the cell progresses to the anaphase. This is all about the regulation of cell cycle. Thank you.